Walking Stick Library presents NIH and NSF Research Turtles. And remember, you can catch us live on Twitch 6 nights a week.
Because I got home fairly late. Anyway, we're going to change tacks. And there has been a lot of talk on Cloud Hub where you don't want to go unless you want to get really challenged and feel a little bit mentally bruised and jangled. I get jangled regularly. I was born jangled as far as I'm concerned. There's been a lot of talk on Cloud Hub about how government agencies are part of the parasite class and civil servants are parasites, and actually there's not much talk. There's mostly a bunch of people posting memes and how they're money laundries with no proof. If you want to prove something like that, you have to show kickbacks and non-competitive bidding, and there's real dishonest stuff out there. They don't show that. It's just stuff they don't want to spend money on. And it's wasteful. So I thought to myself as I walked up the hill, the 4 miles to synagogue, Kramer, what have the NSF and NIH done with red-eared sliders? You own a turtle named Schwa. Little cleanup in aisle 9, folks. Quick bit of cleanup. This is our critter right here. Have they been studied? They are extremely common. They are considered invasive in many environments. They make very good pets. That's about what mine looks like when his tank is clean. This is a painting of one. I like that even better. What we want to do is figure out how to find out what the NSF and NIH have done to pay for research on these creatures. Because why wouldn't you study a common turtle? In fact, why would you? That would be something worth looking at. Let me get 100% here, and I'm going to put the question down here. This is just very basic, unlike most of what we do, much more basic. That's what I'm looking for. And I don't need the or, it won't read it anyway, so I'm leaving out the stop words. I'm not putting it in quotes. I'm going to let Google do all the heavy lifting for me. That's what we're going to do. Let me get Google. And we're going to just paste this in, and I'm going to pull this over, and I'm going to pull this over. And it's Trachemis scripta with an R, Trachemis with a Y and an R. Let's go. Go. Look what I see, NSF Government Award Search. Let's go to the next link. And here we have NIH Health Report. These are search engines that will show you this stuff. Let's make this big enough for everybody to read. And this should be good and big too. We'll start with NSF, and let's do red-eared slider. We can do it Latin, and we can do it in English. And I don't care if the awards have expired. I want to search them by start date. This is relevancy. Financial year 2023, investigating proximate mechanisms and ultimate drivers of temperature-dependent development of male to female ratios in reptiles. Okay. And Andrea Hogada de Regueda got it. September 30th, 2023, she got 30,000 of 240,000. And here we have the name of the person, Breitenbach Anthony, Bloomington, Illinois. He is a postdoc doing biology research. This action funds an NSF postdoctoral research fellowship in biology for financial year 2023, integrative research investigating the rules of life governing interactions between genomes, environments, and phenotypes. The fellowship supports research and training of the fellow who will contribute to the area of rules of life. And here we have the temperature-dependent female or male represents a straightforward case of developmental plasticity in which temperature experienced during a discrete developmental window dictates this development, whereas the molecular and genetic responses to similar thermal cues appear to be conserved. The resultant developmental outcomes vary across species. For example, cool temperatures lead to the upregulation of conserved chromatin modifiers in the red-eared slider turtle and the American alligator, but these temperatures produce males in T. scripta and females in A. Mississippians. My turtle is a male because I think he was hatched in an air-conditioned garage somewhere. They were hatching out a bunch of them and they sold them starving so they wouldn't crap in their water. And this was back in 2010. I've had Schwa for 15 years. He has lived to be lived to be a nice, big turtle, beautiful, full-blown turtle adult. Okay. We know the name of this person, right? I'm going to copy it. His first name is Anthony. And what we can do is we can go to GSU Library and we can search him by author and see if we get him. Go. My guess is he's published. Slow as molasses in January. There, let's open it all the way up. I'm not sure what you would use this for since humans don't have it. Ooh, using naturalistic incubation temperatures to demonstrate how variation in timing and continuity of heat wave exposures influences phenotype. So we're looking at global warming's effect on animals. This is 2000, I don't have a date on this, but we can get a date pretty easily. 2020. So this is a person who studies the effect of heat on animals. Sulfur derivatives of natural polytheoretical arsenicin, a biologically active organometallic, are. I would like to get this guy as an author. So I'm going to open this, and with luck, we can simply click him. This is our guy. Let's do it that way. Okay, so he has published, but he hasn't published anything since. Maybe something is in press. It can happen. Let's do the next one. Okay, so this is responsiveness of sex determination to thermal cues and maternal estrogens as potential modifiers. This makes sense that they're using this organism to study temperature-related sex selection, which probably occurs more in nature than it does in humans. The whole world is not people. Okay, this is Illinois State University, 2023. And Rachel Bowden is the principal investigator. And recent studies have determined that temperature-dependent sex determination involves epigenetic regulation of critical transcription factors that determine sex. In the red-eared slider, Trachobus scripta, also called Trachobus scripta elegans, the histone demethylase KDM6B removes repressive histones marks for the promoter of DMRT1, which is a conserved regulator of testes development. So if it's cool, the histone demethylase turns off the inhibitor and turns on the testy maker. For this species, testy development is induced at cool temperatures below 29 degrees centigrade. So again, that means, I say centigrade, it could be Celsius, I'm old enough. But actually, somewhat useful again. Hello, where is my hair? I didn't even see you, I'm sorry. It was good to see you. Wow, I didn't even see you come in. I was busy trying to get this to run. Yes, it's good to see two people out tonight. How is the art project going? How's the art project going? Did you get it done or are you still working on it? Okay. Let me know and we can throw this in the garbage. Oh, it was a workbook. I thought it was a paper. Oh, I'm glad to hear that. I am very glad to hear that. Okay. When I taught the art appreciation class, they were looking for three-dimensional things. And it all had to be contemporary, post-1950. And we were really good about doing this because we learned how, did a lot of searching in I'm trying to figure out when we first started writing reports when I was in school. I was much younger. Sometimes they're called reports. Okay. Interesting, you're in a high school that teaches art appreciation? That is unusual. Yes, we call it, the names are interchangeable. I don't know where you are, but in the US, the names are interchangeable. English professors would sometimes call papers essays. Papers were just anything you wrote. Okay, so the whole, yeah, my language and your language are not going to meet up. American higher education is very culture bound, and there is a whole, I call it the small paper industrial complex that librarians serve. Students write all kinds of papers inside and outside their major. College is 4 years, and most people come in undecided. They have all these core requirements. Okay. Yeah, we're going to be speaking an educationally different language. And I get, you get to know that. I worked at a college, community college, which was the first 2 years, and it was kind of interesting. I liked the students. They were very smart. They were not well academically prepared. Okay. These are all rather interesting. And again, I wish there was a way to see what, this is an older one. And again, this is a big thing apparently with red-eared sliders, is their temperature-determined sex determination. So here's another one, and this was a long time ago, which is good. I don't care if this was 2003. I like that it was older because I would like to go back and see what this man has written. We'll do one more, and then we will do how temperature both stimulates and inhibits. Okay, so this is more sex determination in red-eared sliders, which is why I think almost all the ones they were selling that night were boys. But I went and bought one. I mean, you didn't get a choice. Okay, this is David Cruz. So let's get him. It's a common enough name, but if we take the name and put it in quotes, we won't get so many. Let's look for him again. I'm just going to throw him in the catalog, throw him at the Discover board. And my guess is we're going to get something. Woo! A review article. Temperature-dependent sex determination and the David Cruz Laboratory. Meets and bounds David Cruz, ancestors and descendants. How the rise of testosterone therapy in men was inspired by lizard research with David Cruz. I want him as an author. You think we can do that? I think we can. This is what advanced search is for. Go. I want what he wrote. Ooh. Is he in here as an author? Let's see if he's here. He is. He's probably not the principal investigator, but temperature can change. Temperature can change reproduction. So it isn't just turtles. Let me grab his name from here because I want him as an AR, not an author. AR must mean author or researcher. That's kind of an odd abbreviation. Passing experiences on to future generations, endocrine disruptors, transgenerational inheritance of epimutations in brain and sperm. So studying reptile sex determination that's temperature induced leads to human health, human reproduction. Two hits to EDCs, those are two hits of endocrine disrupting chemicals 3 generations apart, effect on social behaviors in rats. Temperature shift alters DNA methylation and histone modifications in gonadal aromatase gene in species with temperature-dependent sex determination. So figuring out the mechanism may lead to other things. And of course, there's always global warming issues. And here we have, very recently, epigenetic control of gonadal aromatase in temperature-dependent sex determination of red-eared slider turtles. Litter environment affects behavior and brain metabolic activity of adult knockout mice. Now, knockout, they've taken out part of their genes or turned some of it off. And since this is epigenetic, epigenetics happen elsewhere. Transgenerational epigenetic programming of the brain, transcriptome, and anxiety behavior. So it leads to other things when you are studying epigenetics of turtle sex determination. Okay, let's change this and put in the Latin name, and then this is what kind of thing they're cutting off money from, and it leads to lots of other things. Ooh. And again, this was 2023. They are still studying this, sex determination. Okay, something else I know they study turtles for is breathing. If you watch your turtle, he takes a breath like every minute. If he wants more air, he just comes up to the surface and breathes as much as he wants, and then he goes back down, and if he wants to stay underwater, he comes up every 5 minutes. So they have a completely different way of breathing, and in the winter, they don't hardly breathe at all. Slowing the pace, cellular and molecular mechanisms of bradycardia, that is a slow heartbeat in the anoxic turtle, a turtle without much oxygen in his species, in their cells. And here we have this is at University of Alaska, where turtles will hibernate. This is his name, Jonathan Stesic. I'm going to copy it, and this is how we find if they've ever published. And here, remarkably, the heart, one of the vertebrate champions of anoxia survival. The red-eared slider turtle can continue to beat rhythmically during anoxia, albeit more slowly. Previous research has revealed that a dramatic and rapid resetting of the intrinsic pacemaker contributes to bradycardia displayed by the anoxic turtle. However, the mechanisms by which anoxia modulates pacemaker rate remains unknown. Overarching objective of this research is to exploit the turtle heart as a model to elucidate the physiological and cellular mechanisms of cardiac pacemaker regulation. Gee, I think that might have cardiac pacemaker regulation, might have use in humans. Let's go see if this guy has written anything. Let's see if we find him. We put him in. UBC researcher discovers the secrets of fish that lives without breathing. Ooh, cardiovascular function, compliance, and connective tissue remodeling in the turtle. Trachemus scripta, which is the slider. Trachemus scripta elegans is the red-eared slider, following thermal acclimation. Always know what you own. And I want his name, but his name is not here. Darn it. I do want him as an author. I'll tell you what I'm going to do. Let's advanced search this and find him as an author. Search. I want to find you with no results. That can happen. You have lots of these here, and 3D rotational fit analysis using turtle shells. This was 1991. Good, it was a while ago. F. James, oh my God. Does everybody see the email address here? I almost worked at this library. I job interviewed at Stony Brook. It's amazing what a small world it is. But Dr. Rolfe is at Stony Brook studying turtle shells. What happens with turtle shells? The translation or conversion of shape into quantitative terms so that different shapes can be compared and interpreted is a profound problem in systematic biology. The research will provide information on the application of fitting techniques that will be of use to researchers in many areas of biology on the nature and shape variation of importance to those interested in ecological and evolutionary questions concerning turtles. The proposed research has two main goals. The first is to compare existing and newly developed rotational fitting methods to a set of three-dimensional landmark locations from biological material. The data will consist of coordinate locations of biologically and geometrically defined landmarks on the carapace of the yellow-bellied turtle, Trachemus scripta, and the second goal is to use the data to investigate shape differences resulting from sexual dimorphism, habitat-related growth differences, and geographical variation within the subspecies. That does not work. We're going to try that again, folks. I don't need this one. I do need this one. This is the one which is the one I want? Okay. This is the one I want. There, back to normal size. And let's see if we can find him one more try, folks. Now, this is an old one, which is good. Okay. This isn't him. Ooh. Very nice. And yes, they're doing this for evolution, folks, because when you get back after a few thousand years, there is no more DNA. You have to do anatomical analysis. I used to review paleontology books back in the day. So that's why shape analysis is so important. Let's see if we have him in here. No. He gets mentioned elsewhere, but he's not showing up. Okay. It's a reply to Rolf. So I'm going to assume if we get to the actual article, it'll mention where the papers were published, and then we can hunt him down. This is 1998. There may not be a lot because it's old enough. Maybe it'll mention it at the end. These things usually have references. There we are. Talpidae are moles. Let me grab that out of curiosity. And we can go back and get, I want a GSU library quickly. And let's go. Oh God, this is a mess. Oh yeah, this is a mess because of the way it copied. We just get rid of the garbage and see if this shows up. I may end up with nothing. Oh God, I just saw the mess, or what I didn't do. All right, that's okay if we don't get anything. Try again. We have to log in and we're still gonna have garbage because there are skips. I think we got them all now. Nope, one more. I think we got them all. Let's try. Try that. There they are. I don't know why that didn't take, but there is our author. And he is doing structural, morphometric. Morphology means animal structures. Some of this I know. I was a bio major. Talpidae are moles. I know that from Second Life because the Linden moles have a giant Talpidae, planet Talpidae is part of their sculpture of the solar system in Bay City in Second Life. So we now have an author that we can grab, Rolf F. James. Now that we have him, we can see what else he's done. And this is fun. Not all of it will be okay. So human evolution, again, you get beyond a certain point, DNA doesn't work. Okay, so what we want to do is see if he did anything on, if he ever published the turtle study. Trachomis is the genus name for red-eared slider. Nothing. Try turtles. Look. All right, we've got gastropoda. He's still doing it, apparently. 2012. So he never, the turtle thing never came to light. Sometimes they don't. But we have this person and we know what he did. Okay. Everybody catch their breath. Shall we do the NIH one? And again, you can do exactly the same thing we did before. Red-eared slider, it's a name. We put it in quotes. Who's done red-eared sliders? 4 people have done red-eared sliders. And they're at different places. Now here what they're studying is hypospadias. Hypospadias. Let's get a definition. And NIH is much more human-centric than NSF. Can we go back to 100? Yes, we can. So we're looking for, and I'm going to take this down, I am not pronouncing this well, hypospadias. Doesn't need quotes, it does need to shrink. And we're looking for a definition. Because I think I know what it is, but you don't want what I'm thinking. And we put this together with an and. Fun to color this in. And let's run that in Google very quickly. I'm just moving everything over to where we can get to it easily. Okay, this is the AI top at the, slop at the top. Let's go to the Mayo Clinic. I'd rather have humans put this together. And let's enlarge the image. And yes, this is work safe. That's a penis. And the urethra is open further down. It doesn't come out the end where pee usually comes out. It comes out down here, somewhere along the thing. And it could probably, I always thought of it as slit-shaped. Hypospadias is a condition that happens as the unborn baby, called a fetus, develops in the womb. The opening of the urethra is on the underside of the penis instead of at the tip. The urethra is a tube that drains urine from the bladder out of the body. Hypospadias is common and doesn't cause difficulty in caring for your baby. Surgery can create a typical-looking penis that works properly. With successful treatment of hypospadias, most men can pass urine properly. And with successful hypospadias treatment, the condition won't block sperm from fertilizing an egg. So, yes, if this doesn't get corrected, the little boy goes sitting down. Because he dribbles, and he's got to wipe really well. So, here is the nature and systematic mucosal function of B cells in reptiles. These are, and this is hypospadias morphogenesis. How do people get hypospadias? Because if we know how they get it, we might be able to prevent it And they are doing it in a turtle, apparently, as a test organism. We'll eventually get there. Hypospadias is a congenital defect characterized by an ectopic opening in the urethra on the ventral side of the penis. It is one of the most common birth defects with an incidence in 1 in 125 live births. Currently, the only treatment for hypospadias is surgery. The high incidence of hypospadias is reflective of multiple causes. And down here we have what they use the turtles for. Aim 2 will be to address the molecular control of poomorphogenesis through examination of changes in gene expression and cellular events in the BMP2 and FGFR2 mutants. Both BMP2 and FGFR2 are expressed in the puo region, and the FGFR2 mutants show defective poomorphogenesis. Finally, aim 3 will be to examine the cellular and molecular mechanisms that lead to a naturally occurring open urethra in the red-eared slider turtle, Trachomas scripta. This aim will allow for the identification of a novel mechanism important for manipulating urethral closure. Findings from this proposal will not only add to the knowledge of how external genitalia normally develop, but also identify mechanisms that lead to the generation of hypospadias. We might be able to cure this better than prevent this. That's why there's so much research on it. And here we have B function in reptiles. The reptiles are a long-lived vertebrate group often overlooked in immunology research. Hormonal immune system of reptiles contains both innate and adaptive components. Fun things about your reptiles. Oh, you're both here still. Thank you. I haven't been checking my chat like I should. The humoral immune system of reptiles contains both innate and adaptive components, but the organization of these components differs. For example, reptiles lack lymph nodes, but have gut-associated lymphoid tissue. In addition, researchers are just beginning to appreciate the nature of the humoral immune response, particularly B-cell function in reptiles. Given the many similarities of human and reptilian humoral immune system, it is surprising that long-lived reptiles appear to maintain immunocompetence throughout the aging process, unlike humans. Wow. Why? Because I own one. I have a turtle, and it sounds like useless research, right? A bunch of people are saying NSF and NIH is useless, and they've cut off funding for a lot of the grants. It's very scary. We're about to kill our science in the United States. We have a crazy government. We do. I mean, people who elected him because they didn't like Biden didn't elect this. They got stuck with it. Actually, turtles are good models for a lot of things. They're good models for sex. Surprisingly, studying turtles with your tax dollars pays. That's the point tonight. As somebody who owns one, I can tell you that they are good creatures to own. They are not social. That just isn't part of their brain. But cats are social. I have 2 cats and they're incredibly social. But the turtle is not social, but he's not stupid. And he has his likes and his dislikes. He puts up with a lot from me. He's the only creature I've ever been able to raise from infancy to adulthood that has lived a long time. Of course they are. I mean, if you've ever they make good pets. And in this country, it is illegal to buy a red-eared slider hatchling, although I bought one because they were selling them at the fireworks 15 years ago. I don't know if they're legal in your part of the world. And certainly what I have is invasive in your part of the world. You've got to really watch. Someday we will do cats in New Zealand. If you're around where is my here, and you want to do cats in New Zealand, we will do them because they aren't invasive. And there was a big controversy about culling cats. Now here, well, they're invasive here too, but this is a big continent. And they also kill vermin, so people like them. They even like the feral ones. They put them in barns and places and stores in places where they have a job to do. Wow. So anyway, they did this, and they would reptiles keep their immunocompetence when they age. Humans do not. So if we study reptiles, in this case turtles, because these are a common creature, I would think they'd be difficult to study because of all the bones. If we study turtles, one of the things we will learn is more about our own immune system and how to make it better. This is what primary research does. And let's just take a quick look and see what happens with Trachemis scripta. In fact, I'm just going to use Trachemis. I'm not going to worry about the subspecies. Ooh, why is this involved? Here they're doing one in ears. Cochlea is in the ear. Ooh. There's a model of aging. The cochlea is in the ear. The goal of proposed research is to characterize the cellular mechanisms that contribute to tuning in the cochlea, using a combination of electrofluoroscopy and fluorescence imaging techniques, both on solitary hair cells, that's in your ears, folks, from the red-eared turtle Trachemis scripta elegans. We will address two questions. How does the expression of channels that determine characteristic frequency vary along the length of the epithelium, and how is the quality of resonance modulated by efferent fibers and mechanical properties of the hair cell? These are nerve cells. These are cells that lead to nerves. This is how you hear. In answering the first question, we will test the hypothesis that tuning in lower frequency cells is due to an assemblage of voltage-dependent potassium conductance, partially adapting outward rectifying conductance. By understanding how these cellular mechanisms determine frequency selectivity, we will be able to assess their limitations and applicability to physiology and pathophysiology of hearing in higher vertebrates, including man. And of course, losing hearing is a big thing. People becoming hard of hearing is a big thing. So again, no waste of money. Oh, they're invasive everywhere. They're native to here. In the United States, these kinds of regulations are usually state level. And I don't know how they got away with selling them. Joie is a gray market turtle. I'm kind of glad I got him. He was a perfect fit for me. I wouldn't give red-eared sliders to little kids because they can get mean when they're abused. And they can bite very hard. I've been bitten twice, and I keep my fingers well away from his mouth. He can claw me, but he can't bite me. And he doesn't know how to modulate a bite to say, no, human, get away. I don't want to kill you, but no. Ozora, my cat, and Emmanuel, my other cat, if they really want me to say no, they'll stick out their claws just a little bit and hit me. Chances are good they won't break the skin, and they'll get their message across. I'll feel the claws, but they won't scratch. So cats have the ability to modulate a blow. Turtles do not. And cats are social. So this is actually pretty interesting. I didn't think they did all this. And here again, they do oxidative stress, immune system, a lot on hearing. Um, and here we have brain reoxygenation. So people study turtle. Your tax dollars in the U.S. go to study turtles. They aren't wasted. There's lots of esoteric stuff people study where the money is not wasted, because ultimately it goes for something that we can use. It may be 2 or 3 studies down the line, but it keeps these people in business. They support the local economy while they're in business. They go to the beer store. Sorry, Tinker, had to say that. They go to the beer store. They go to, yes, in New York there are beer stores. Here in Atlanta there are package stores. Different cities. He's in New York, I'm in Georgia. We're both in the eastern U.S. They pay rent. They buy things. They pay taxes. So when you fund research, you're funding the community where it happens as well. I'm a little unhappy tonight because we have 4 families that work for the CDC that I know of in various synagogues congregations. We have 3 in my congregation. We have an IRS and 2 CDCs in my congregation. We have 3 families that work for the government. There's another family I know where the husband works for CDC. So that's 4 families at least, and there's probably a heck of a lot more. And we're effectively looking at what happens when they close the steel mill and kill off a bunch of jobs. And it's very strange to be going to the synagogue and knowing that the community around you is going to face a financial crisis, because a lot of our membership is middle-class people with government jobs. And these are families with young kids because they could afford to have them when times were good. And now they're going to be in over their heads. It's just not very good. And our rabbi is not being responsive. He wrote about something deeply personal and read it to us. And a lot of people liked it. I didn't know the person he was writing about, and I care more about what's going on in Atlanta than what's going on in Israel. Sorry. That may be heresy, but we as a community have to plan to be doing some pastoral support. We need to get our boots on. That's not what you want to hear tonight. You guys have been so stinking patient. Hello, everybody. Just making sure you're all still here. You've all been really stinking patient. And again, we can see if anybody wrote any of this. Really quickly, let's check one more author. Larkin's Christine E. Okay. Let me get a GSU library up. And we can look for Larkin's Christine E. Our person who studies hearing. This is a university with a 4-year American Sign Language program that I'm looking for articles in. So this is, again, there she is. Actually, this is the one about the, wow. There it is. Spatiotemporal dynamics of androgen signaling underlie sexual differentiation and congenital malformations of the urethra and vagina. But let's click her because we want her as an author. We'll see what else she's written. So she is a genetic researcher. Sonic hedgehog is a gene, and people hate that it's called that. I think it's great because it's a lot easier. I think it's a lot easier to pronounce than many of the other genes. and easier to remember. So, a lot of stuff about fertility and congenital malformations. And she's done a lot of this. And again, we could see who cited her, and you can follow the research. There she is. They are using them as a model for human development. And phallus is penis. Okay, and I can say that with a straight face because it's an animal part. All right, let's go back inside. And I'm going to put my curtain back up because you don't need to see me reflected 20 times. That's not fun. That is not fun. So we have a stage here. So point was tonight that the NSF and NIH don't waste your money. Thank you so much for watching. And don't forget, you can catch me live on Twitch.